Hello, welcome to demola. In today's lesson, we are going to be talking about cell membranes. A very important foundational topic as we begin topic 2 now. Let's get straight into it. Okay, so we cannot talk about cell membranes without mentioning phospholipids. These are going to be the most important part of your cell membrane, yeah? So, when we say phospholipids, this is what we're showing, yeah? If I circle it in blue, that is a phospholipid, yeah? And it's made up of two parts, the phosphate head and the fatty acid tails. We're going to get into more detail about both of them, but I just wanted to show it on a diagram. We've got the circle, the phosphate head, and the two lines, the fatty acid tails. And what I also wanted to mention is that they do have glycerol as well. So we don't show it on the diagram just because functionally it's not as important in the phospholipid compared to a triglyceride, for example. But it is there. It's going to be in between the two. Okay. Now let's talk a bit about the phosphate head. This is hydrophilic. Hydro, water, philic, loving. Yeah? And it gets that property because it is polar. What does it mean? It means that it's attracted to water. It's soluble in water. Yeah? And then if we talk about the next region, the fatty acid tails, hydrophobic, water, and then phobic, hating. Yeah? So this region is non-polar. It's repelled by water and soluble in water. Either way, this one does not like to relate with water. So that's important because we form a phospholipid bilayer now. The phospholipid bilayer is basically the cell membrane, yeah? Everything in the cell membrane works around that phospholipid bilayer. So, just to go into a bit more detail, extracellular matrix, the fluid outside of the cell, contains water. Intracellular matrix, the fluid inside of the cell, also contains water. Hence, your hydrophilic phosphate heads are going to be attracted to it. Yeah, it will face towards it. Whereas your hydrophobic fatty acid tails want to face away from it, so they face inwards towards each other. Now, we need to go into a bit of detail why that's important in terms of transport across membranes. This will be elaborated on in a later video, but for now, if we have a small non-polar molecule like water, for example, yeah, water is going to have a great time getting, oxygen is going to have a great time getting through because it's small, so it passes through the phosphate heads, and it's also non-polar, so it's able to get through this hydrophobic region and come out on the other side of the membrane. Yeah, happy days. Whereas, say we have a large polar molecule like glucose, for example. First barrier, glucose ain't going to be able to pass through here. No chance. Glucose can't fit through there. So that's one reason why. Now, let's maybe switch it up. Let's do a small polar molecule like, for example, sodium. A sodium ion, yeah? This is relatively small. It's polar. So maybe the sodium ion can pass through here, the first line of defense, but no chance it's getting through this hydrophobic region, yeah? No chance. So because it's a polar molecule, can't get through that hydrophobic region, hence, that is how your cell membrane has a bit of permeability by itself. We are going to go into more detail about that, not only in this video, but in the next video when we do transport across membranes. So look out for that. But for now, this is what we need to understand about phospholipids and their role in the cell membrane. So in this lesson, a lot that you'll see is us building up. So we've got a phospholipid bilayer here, right? You can see the phospholipids, you can see the phosphate heads facing outwards, the fatty acid tails facing inwards. But there's also a lot of different molecules that are in and around our cell membrane as well. Let's name them. We've got glycolipids, yeah? We've got cholesterol, intrinsic proteins, extrinsic proteins, glycoproteins, yeah? You just need to be aware of these things are going to exist in your cell membrane as well. Now, let's go into a bit more detail. A glycolipid, yeah? Literally, glyco, sugar, and then lipid, obviously lipid, yeah? So it's a lipid attached to a sugar chain, yeah? This one's involved in cell signaling and membrane stability, which a lot of them are involved in. Cell signaling, you can say that in exam. You can say cell recognition in exam. I would mainly go with receptors, yeah? So those are kind of your features of what they can do, yeah? Glycoprotein is going to be essentially the same. Glyco, sugar, protein, yeah? A protein attached to a sugar chain, yeah? Same thing, cell signaling, membrane stability. You can say recognition, you can say use as receptors, yeah? Now, extrinsic proteins, again, quite similar, but we're going to make one distinction, yeah? So you see this extrinsic protein, it's not going to be embedded in the membrane. Oh, it's embedded in the membrane, but not across it. So intrinsic protein, you want to say embedded across the membrane. Extrinsic is just embedded on one side of the membrane, yeah? So, for example, the one here is, let's say that that's the extracellular side, yeah? What can this do? It can do that cell recognition. It can be a receptor, yeah? This can also be an enzyme. So just be aware of the different things that can do, yeah? What else did we want to talk about? We wanted to talk about intrinsic proteins. Now, intrinsic proteins, you can see they span across the width of the membrane. Sorry about that. They span across the width of the membrane, yeah? Mainly going to be transport. Cool. So channel proteins, carrier proteins, we're going to learn about that more in transport across membranes, but just be aware of those terms for now. And finally, cholesterol, which membrane stability, yes, but I also want to mainly focus on it in terms of membrane permeability. If we have more cholesterol, that will affect the permeability of the membrane. So just be aware of that as well. Now, those are kind of the key components that you're going to find across the cell membrane. Okay, and something else I wanted to mention about cell membranes, just because this is mainly just building foundation so that we can actually talk about transport across the membrane. I just wanted to refer to models. So the current one that we use is Singer-Nicholson or the Fluid Mosaic Model. Yeah, and you can kind of see it there. It's kind of what we've already drawn earlier, just a bit in more detail. But why is it called Fluid Mosaic? Well, fluid. Something that I haven't mentioned is that contents can move freely across the membrane. Not across, but along the membrane, yeah? So, I mean, if it's on the extrinsic side, it's going to be moving across there. If it's on the intrinsic side, it's moving across there, yeah? This will be your phospholipids, your proteins. Either way, they can move transversely or sideways along that membrane, yeah? That's going to be something that makes it fluid. Now, mosaic. I'm not sure if any of you have seen a mosaic protein, like a mosaic art piece, yeah? But it just means there's so many different components and all dotted around. So it kind of looks like that. I mean, if you look at it here, you can kind of appreciate that there's kind of a random arrangement of just different things. If it's glycolipids, glycoproteins, extrinsic proteins, etc., they're just kind of dotted all over the place. So it gives it that kind of mosaic vibe. Now, fluid is really important just because it's going to help you, again, about getting things across the membrane. So endocytosis, exocytosis, these will be covered in later videos, but that involves the membrane having to be flexible and dynamic, which is going to help substances to get in and out of the cell. Yeah? Mosaic, it just means you can have a bunch of different components there. Now, we also have the Davson-Danieli model. Yeah? So this is an old one. Let me just highlight something so that you can know which one we're talking about. Talking about this guy here, the Davson-Danieli model. Yeah? They also called it a sandwich model. So essentially, they're saying that the proteins form distinct layers, and this got disproven. Yeah, it got disproven that this isn't the actual model because, for example, how are your non-polar things meant to get through? If there's just a layer of just these proteins, yeah, no chance that a molecule can get through. If it's polar or if it's non-polar, you're gonna struggle to get through because if it's polar, it can get through that, can't get through the hydrophobic region. If it's not, because there's no protein spanning across the membrane, right? If it's non-polar, can't get through the polar region to get into those fatty acetals, yeah? So you can see how that model got disproven, right? Something that you need to think of, and I don't have any notes of it in this video, but just be aware of this. We need to think about development in terms of science and how things get proven, disproven. Yeah, how theories kind of originate. Things that you're going to want to say if that comes up. It's a niche question, which is why we haven't focused on it. But you can say things like microscopy, for example. They would have seen that cell membranes don't look like that. Yeah. Potentially it could be experiments. Yeah, they could have done experiments that prove, oh, hold on a minute. This substance can get through, but my model doesn't show that that can happen. Yeah. It could be peer review. Anything like that. Just make sure you're mentioning things like that when it comes to scientific studies or like development. Okay, and it is question time. So we've got a multiple choice one there. Which of the molecules found in cell membranes have hydrophilic regions? Answer that. And then we've got a bit of a longer question here. 2 parts. Have a go. We'll pause and get the answers up for you. Okay, and here are your answers to that question. Now, with the first one, I think the only thing that would catch people out is the fact that cholesterol is here. Yeah. Now, let's kind of talk about this. You see cholesterol, how it sat on the outside of the membrane, but then also some of it went inside. Not the whole way, but it went inside. That dictates to you this must have polar and non-polar regions. Yeah. If it can be outside with the polar bit and also be inside with the non-polar bit, it should have both. Cool. Phospholipids we talked about in enough detail. I think we'll find out. And in terms of your channel proteins, To be within that phospholipid bilayer, as you would have seen in the question, the outer regions must be hydrophobic so that it can exist with those fatty acid towels. Yeah. But then what you'll notice is inside, so that we can allow the passage of ions and water and everything like that, again, will be covered in the next video. To have that, we must have a hydrophilic region in the inside so that those polar molecules can pass through. Cool. Let's talk about this one. We mentioned this in detail. We talked about this in detail. What is the function of, I think it was glycolipids or glycoproteins, yeah? Cell recognition, receptors. This is my go-to, receptors. Antigens as well, I think is a good one to mention. Just make sure if you are talking antigens, this can only be glycoprotein, yeah? It can only be a glycoprotein or an extrinsic protein. So don't say that if you're talking about a glycolipid. Now, last question. We've talked about this in detail, yeah? The phospholipid, what does it have? We'll highlight it. It's got the polar phosphate head, and it's also got the non-polar fatty acid chains, yeah? Mention both, gets you a mark. We talked about, because of the fatty acid chains being inwards facing towards each other, that only these non-polar molecules can pass through, yeah? And the polar ones cannot. So just be aware of that. That's an answer related to all of the theory that we've covered in this lesson. Okay, so it's just one of these ones. We're on to the A-star tips. Let's get out of the way. We can focus on transport across members in the next lesson. So you need to understand the phospholipid bilayer. Very, very important. In fact, probably the most important part of this lesson is that you know what the bilayer looks like. What are the components of a phospholipid? Yeah. Fluid mosaic model. I want you to be able to describe why it's fluid, because things can move. Why mosaic? Because of the random arrangement of things in the membrane. Yeah. Those are easy marks to pick up if they come up in an exam. Yeah. Know the functions of other components of the membrane. Very useful to know as well. You saw the question now where they asked about glycoproteins. Yeah. Very useful to know what the functions of those components are in case it comes up. Now, what do we not do? Not knowing what can pass through the membranes, guys, we are going to cover this in the next lesson, transport across membranes. We need to know which processes allow things to come across the membrane. Hydrophilic, make sure that you never forget it. I kind of think of it like phobia, fear, right? So if you're fearful of it, you don't like it, you hate it, yeah? That's how I remember that hydrophobic means repelling or it hates. Hydrophilic means it loves. Cool. Don't neglect the old model, Dawson Danieli. I wouldn't say this is the most important thing, but definitely can help you to gain a mark of 3 here and there if you know the other model. Not saying it will come up, probably even unlikely to come up, but in case it does, you don't want to get called out by it. Cool. That's the end of that video. So a lot of the work that we did here was so that we can talk about transport across membranes in the next biology video. But I hope this was useful to get an understanding of cell membranes. Like, subscribe, ask any questions in the comments below. I'll do my best to answer. We got the TikTok and the Instagram, both at A-star demola, and I will catch you in the next one. Bye for now.